Okay. So sort of the main focus of this lecture is statistics and reproducibility. And so this is really trying to cover a lot of the ideas that we've sort of mentioned before in this class, but bring them all into how do you actually take an experiment that you're performing and run a meaningful sort of analysis on it. So there's a fairly large amount of additional reading that goes with this. I think this is one of the most important lectures to really understand. I assume most of you have taken statistics classes before. Has anyone not taken any statistics class? One person, okay. So I think everyone else probably has taken statistics. I don't necessarily think it's a requirement because I think most statistics classes aren't that well taught. I don't necessarily think this is brilliantly taught either, but I try to make it a bit more understandable and at least reference more practical uses because it is probably one of the most important things you'll do, particularly if you're a PhD student. For master students, it's obviously important as well, but if you're doing a PhD thesis, statistics is something you should really have a good feeling for because that's fundamentally your experiments and how you know sort of what you did. So here we've got sort of a number of videos and podcasts and articles that try to kind of make this easier to understand. So of course there's Google and Stanford statistics lectures, which are brilliantly done. There's very nice podcasts available where they talk about sort of t-tests and p-values, which I find very helpful to listen to. Videos about sort of correlation and causation, as I'm sure this is something many of you have heard before. And then kind of the last two are really focused on the credibility problems. And so this is why it's so important for researchers to understand these topics, because this is sort of the basis for what science and the scientific method is built on, and there are huge problems with a lot of the research that's being done today. And sort of understanding how this works, what's problematic, what's not, is quite important. I then have some more sort of slightly less serious links here. So the first one is sort of how to solve natural language processing problems. And so it's obviously a very different topic than what we're covering, but it's a quite nice slide set covering, so if you click on it, you get to the tweet, which is then on Google Drive, and this is a professor for computational linguistics, and it's how to find and answer a research question. And so it's really very, very sort of fundamental material for understanding different sort of problems, how you design an experiment, what sort of data you collect, how you do a literature review, what's a hypothesis, what do controlled experiments mean, what is a baseline, upper bound, so things that we've certainly mentioned before, but covers quite a bit of, sort of specific topics with natural language processing. So it's not obviously completely transferable to the image domain, but I find the slides quite useful and very well written. And so they explain a lot of the things potentially better than we do. The second one is sort of data visualization, which I think is incredibly important for sort of understanding what you're looking at and then presenting to other people and getting sort of a clear idea of what's going on. There's a number of resources that have more on this. P-values with puppies. So this is sort of a slightly less serious article, but I think it's from the chief data scientist or some title like that at Google. So someone who's obviously dealt with lots of data and understands the importance of statistics and trying to make these complicated ideas like p-values much more tangible. So the math of calculating a p-value or a t-test is very easy, but really understanding intuitively what it means is quite a bit more time consuming. And then examples, so just Stripe is a financial transaction company, and so they have to deal a lot with sort of statistics and reproducibility in their analysis and how you process all this information and why it's important. And then sort of the last ones are a bit more on the sort of reproducibility side and repeatability. But that we won't go into as much detail now. So kind of previously on this course, we did a lot of kind of the image enhancement, the segmentation technique, shape analysis, complicated shapes, the dynamic experiments, and sort of the idea from all of this is that we've built up a set of tools that we feel comfortable with a number of different types of problems. So the first lectures were really to try to give you a tool set so that when you have an image problem, you can start applying tools to it to at least make the problem easier. You probably can't solve any problem you come across, but that a lot of the problems you run into, you can at least make inroads on. And so again, kind of as we went into last time, the course has been called sort of quantitative big imaging, but what does big imaging even mean? With dynamic experiments, sort of that became quite clear that we were looking at sort of lots of different data points and we wanted to analyze them in a similar way. What we're really focused on for this lecture is sort of, doing analyses in a disciplined manner. So sort of fixed steps, easy to regenerate results, no magic. Having everything automated, being able to adapt and reuse analyses. And so what we're focused on for this is really that whole reproducibility part. So how do we make sure the analysis we do are able to be reproduced, and how do we evaluate that well? And so if we kind of go back to one of the first lectures in sort of objectives, that the goal of scientific studies, quantitative scientific studies, this might be slightly oversimplified, is to get a single number. And so all of the tools that we've covered up until now are trying to result in numbers. In some cases, maybe a field of different numbers, but in most cases, we'll be trying to look at just a single number. And so you're assessing, the average flow rate over 100 frames is, 1 millimeter per second, or the number of particles is 97, or the size of the particles is 300 cubic micrometers. But all we do is try to get one number out of our analysis. And so what we've done before is make sure this number is sort of describing what we're trying to look for in the image well. And so how do we quantify structures, shapes, changes with this number? And today what we're trying to do is make sure this number is meaningful. So as we covered in other lectures, you can do sort of complicated curvature analysis, have Gaussian curvature over all of the surfaces, but that doesn't necessarily mean that that number has any significance. And so today what we're trying to do is bring that kind of analysis with simple examples into, what can we actually do to ascertain if there's differences in what we're looking at. So that sort of specifically translates to how do we compare different samples in groups. And so when we make a number of different measurements, how can we go about and start to show if these samples are different or if these samples are the same? And then how do we start to sort of compare different parameters, filter choice, minimum volume, all of these pipelines and analyses that we've come up with have a large number of different parameters that we need to sort of tweak and analyze. And how do we sort of ensure that the techniques we have built do what they're supposed to do? And then the kind of last part is how do we visualize so much data? And sort of simple rules to follow for doing that. So kind of the outline, we'll start off with sort of reproducibility with some sort of statistical metrics, then some things about sort of parameterization, unit testing, and visualization. And so we'll try to keep it fairly focused. The predicting and validating, we sort of already covered in one of the earlier lectures, so we'll have some slides on that, but we'll try to keep that to a fairly minimal amount. And so, if we start off with kind of our original cell image, we got rid of noise, we analyzed the shape of the shell, the cells, We then, separated them with watershed. We could track them. We could do a whole number of different sort of analyses with it and calculate a huge number of metrics. It's potentially, interesting that we could do that. Those are the tools we've covered now. But how do we really move on to do something meaningful with it? So this is where we kind of get to this correlation and causation. Switch back to the that I presume most of you have heard that correlation and causation are not the same thing. This is probably one of the most important core ideas in statistics, where, and most common criticism of scientific work is that you're sort of confusing correlation with causation. And sort of correlation means that there's a statistical relationship between two things that you look at. If you wake up today and it's sunny and it was rainy yesterday, and you've done that 10 times, then maybe you think after it rains, it's sunny. That's correlation. It's fairly easy to show. You can just take a number of measurements and show that it's kind of reasonable. And causation is a much more sort of high bar to reach. And so really what it implies is that there is some sort of mechanism between A and B. And so correlation, you could say, the number of cookies I eat is correlated with my weight. And, the correlation could be one direction or the other, that, the heavier I get, the more cookies I eat, or the more cookies I eat, the heavier I get. The correlation doesn't imply any directionality, but the causation, of course, does, so that there's a mechanism between A and B. And so it's much more difficult to show and sort of impossible to prove. And so with these two ideas, it's quite important that you sort of keep them separate and use the correct term at the correct time. And so if you've just done some experiments and analyzed your data, all you can really talk about are sort of correlations. And it's only when you've sort of taken the next steps that you can get into talking about causation, but you can never talk about causation with the same certainty that you talk about correlation. And so one of the major ideas here is this idea between kind of controlled and observational. And so there's really two broad classes of scientific studies. So the observational is kind of the much more popular today, and controlled are kind of the experiments that youth did in high school, that sort of the original fathers of science performed in order to sort of understand the world that we live in. So the observational are much more of like the Facebook, the big data, the Google, where they have sort of huge data sets that they're looking at, and they're kind of trying to find trends and correlations and patterns. And you sort of have a population that's fairly random, and you're not really hypothesis driven. When Google built their search engine, they were trying to make the best search engine that they could produce so that you could search through content very quickly and easily. When they then used this to try to predict when flus outbreak, that wasn't what this was made for. They were taking a search engine that was meant for searching and sort of applying it to other use cases. And so what sort of problem with this is it sort of rarely leads to causation. And so it's a very, very useful data set. It's very good for kind of understanding trends and for suggesting hypotheses, but it doesn't really give you the same kind of scientific evidence as a controlled study does. There are certainly circumstances where it can, and there's certainly lots of circumstances where you don't really have any other way of doing that kind of experiment. If you want to, experiment with, does a vaccine work well, it's not really ethical to tell 20 people that they shouldn't use vaccines so that you can see if their kids get sick and die. It would be the only way to do a controlled experiment, But because we're fairly confident vaccines work, you wouldn't want to have a controlled experiment where you take someone and intentionally make their life more difficult in order to show that there's actually a difference. Obviously, before you knew vaccines worked, it would be quite easy to say, we have 10 people that we give the normal treatment to and 10 people that we give this new vaccine, and we see which one works better. But with a lot of things, once we know that they work and once we have some confidence in that, it becomes very quickly problematic to not give the best treatment you're aware of to people. So controlled is really the most scientific studies, at least traditionally, fall into this category. The specifics of the group are very strongly controlled, and this is much better evidence for causation. And so when you're actually taking the groups, you're controlling what they do, you're controlling what happens to them, how you separate them, how you define them, you're in a much better position to isolate all of the other different possible factors and try to show that there's actually sort of a difference between these two groups. And so if you kind of look at these example experiments, we examined 50 mice with gene XYZ off and 50 with XYZ on, and the foot size increased by 10%. And so if you did that, and that was the only difference between these two mice, or the two groups of mice, then you could start to get into causality because you've really tried to control this group. And if you look at the same kind of problem, with a big random group of mice, so if you had, 1,000 people and 500 had XYZ gene and 500 didn't have XYZ gene, and the ones that did have it had a slightly larger foot size, that would be much harder to do something with because you don't know anything about what other genes they have, what environments they came from, what their backgrounds were, if they wore shoes or not, all of these other factors, which then make it sort of confounded so that you don't know which possible change was responsible for the outcome. Whereas with these controlled studies, you have sort of explicit focus on just getting these two differences and then looking at that. And so, we increased the temperature and the number of pores and the metal increased by 10%. And so you have the exact same structure, the exact same synthesis process, the exact same oven and everything else, and all you did was change the temperature, and that, led to a pore increase. And so there, because you were controlling so many of the other factors, you were able to do that in a much better way. And so these two concepts are quite important because a lot of times when studies are done in an observational way, they're almost presented as if it was sort of a controlled experiment, and they are very different and kind of much different in how you're supposed to analyze them as well. And so, of course, the advantage the observational have is that you can use much, much, much larger data sets because you don't need to control what everyone does. And so, an observational study with 100 million people, is potentially more valuable than a controlled study with 10. But they're still coming from very different standpoints. And so if we kind of take a simple model of kind of a magic weighted coin, where you sort of buy a coin at a shop, and how many times do you need to flip? So basically the magic coin is that you get a coin that is supposed to be heads more often than tails. And so you buy a coin at a shop, and the kind of first question that comes is sort of how many times do you need to flip it to prove that it's not fair? Infinite. Yeah, well, that's the right answer. So if you flipped it 10 times in a row, and it was 10 every time, or it was heads every time, sorry, then that would seem unlikely, but it's not impossible. You could flip a normal count 10 times in a row, and it could get 10 heads. And so it sort of becomes a problem of, how do you decide what fair means and how do you decide if you've proven something or not. And so if it's really unfair, so if it's 100%, then you probably don't have to flip it that many times to show it's fair. If it actually gives you heads 52% of the time, then you have to flip it a lot more times in order to show that it's actually fair. If I flip it 10 times and another person flips it 10 times, is that the same as 20 flips? Yeah. 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 So that it's not the same as if you were to flip it 20 times. And so if you have multiple people different doing the task, and with coin flipping, you wouldn't expect it to make that much of a difference, but there are people who flip coins in very strange ways that might cause the magic coin to be sort of more biased or less biased. And so it's not the same as doing 20 flips. If I flip it 10 times and then multiply the results by 10, is that the same as 100 flips? I mean, certainly you've seen people do this in chemistry experiments, where they do 3 measurements and then they just take the column in Excel and drag it all the way down to get what the rest of the measurements would have been. And your teacher told you not to do this for sure, but why? Yeah.
Just a copy of your results. You do not generate extra data, you just copy or duplicate your data, so you don't get extra information.
Right. Right. So the problem is, yeah, you just copy it. And so with, if you flip a coin twice, you could get 2 heads. That's not that unlikely. And if you multiply that by 10, you would now have 20 heads, and that's extremely unlikely. And so the sort of little biases that cancel each other out as you go out don't cancel each other out if you multiply. And so it's sort of the difference. And then the kind of ultimate question is sort of when you buy 10 coins and you want to know which ones are fair, what do you do? And so what this kind of comes down to is sort of fairly weird letters, but fairly simple math where you basically say, each coin represents a stochastic variable, each flip represents an observation, and the act of performing a flip is an observation on this variable. And so that the sort of the GI is equal to F of G. And what we say is that sort of a fair coin has an expected value of 0.5. So that every time you flip it, you expect half of them to be heads, half of them to be tails. You also sort of say that you probably, in most cases, have an unbiased flipper. So clearly, if your friend flips the coin in a very strange way, then that's not true, but that, sort of each flip is independent of all the other ones. So that, the fact you flipped 10 doesn't make it less likely or more likely that you'll flip a heads the next time. And that each person who's performing the flip has more or less the same result. And finally, that the expected value of the flip is the same of that as the coin. And so really what that means is that you don't really have any temporal dependencies. So if you had a coin and once you flipped it 10 times, it's warmed up, and then it's much more heads than tails, that sort of breaks these standard assumptions, which make it a lot more difficult to do this experiment. It's still possible, it's just different statistics that sort of get involved. And so what we kind of see is that, we have this simple model, which is the coin flip, and we have these real experiments. And for most of you, the link between what does flipping a coin do and what is a real experiment is probably not very clear. And so really what we have with this coin flip is that each flip gives us a small piece of information about the flipper and about the coin. And that's sort of why we can't multiply the results by 10, because we don't get 10 times as much information by doing that. We only get that by performing more experiments. And so the more flips provide us with more information. And kind of the main things here is that random and stochastic variations in the coin and the flipper start to cancel out. And at the same time, systematic variations accumulate. And so, if you have a biased coin that's heads 60% of the time, the more you flip it, the more present this bias will be, because that's sort of a systematic variation. But if you flip a coin twice, the likelihood you get two heads is very high. If you flip it 100 times, the likelihood you get all heads becomes very low. And the same thing sort of occurs with real experiments. And so each measurement tells us more about our sample, our instrument, and our analysis. And more measurements provide more information. And the first line there is quite key. That's the same with the coin flip, is that you're not measuring anything independently. So with a coin flip, if you have someone who gets 10 heads, you can't easily say if it was how they flipped it or if it was the coin. You kind of have everything mashed together in just that observation that you're getting. And in real experiments, it's the same way. So you're learning a little bit about your sample, a little bit about your instrument, and a little bit about your post-processing analysis, but it's all tangled together. And it's quite difficult often to separate all those different components. And so more measurements, again, give you more information, and that sort of random and stochastic variations in sample, instrument, and analysis tend to cancel out. And sort of normally, the analysis has very little to no stochastic variation. Some of the methods that are sort of more complicated in segmentation do, but typically that has very low. But systematic variations accumulate. So if your threshold in your analysis is too high or too low, the more experiments you do won't get rid of that problem. It just makes that problem more and more present. Similarly, if your instrument has the wrong offset or bias, that won't cancel out with more and more experiments. And so now I think we can take a quick break and then get on to a more complicated model, unless there's any questions on this.